Hello students, welcome to the biology class. Today we are going to continue with our same chapter, Biomolecules. In our previous session we discussed about the macromolecules, and today we will discuss the macromolecule, which is your proteins. What are proteins? Proteins are heteropolymers containing the chain of amino acids. A peptide bond formed between the carboxyl group and the amino group of successive amino acid joins the amino acid together. When about 50 or more amino acids are linked together, the molecule is called polypeptide, which is known as protein. So what are proteins? Proteins are the heteropolymers containing the amino acid. Amino acids are known as the building blocks of the protein. A peptide bond is formed between the two adjacent amino acids, that is between the carboxyl group of one amino acid and the amino group of the adjacent amino acid. And the bond which form between the amino acid is known as the peptide bond. And the linkage of that peptide bonds or the amino acid through the peptide bonds, that is known as polypeptide. And the polypeptide, polymerization of polypeptide is known as the protein. Although there are just 20 different types of amino acids. So as we know that we are known with only 20 amino acids, but these 20 amino acids, they form the thousands of different types of proteins. So how that is possible? That depends upon the number of amino acid and the sequence of the amino acid. For example, we name the amino acid A1 to A20, and if one polypeptide is of A1 to A5, another is from A1 to A7, so that becomes a different types of proteins. If we are having A1, A2, A3, A4, 5, but in another we are having A1, A5, A3, A4. That means the sequence is different. So with the change in the sequence, we will get a new amino acid. Similarly, in English we are having 26 alphabets. By the arrangement of that 26 alphabets and the number of that, we will have an infinite number of the words in our dictionaries. Similarly, with the number or the sequence of 20 amino acids, the arrangement of 20 amino acids and the number of 20 amino acids, we can get the thousands of the protein, and they are different from each other. So the next is structure of proteins. So proteins shows 4 levels of the structure. First, that is primary, secondary, tertiary and quaternary. So first we will discuss the primary structure. The protein molecule in primary structure is linear polymer of amino acid, that is a straight chain of amino acid joined by the peptide bond. Primary structure of protein is defined as a sequence of the amino acid in the polypeptide chain. Such polypeptide is non-functional. In this chain, amino acid at the left end is first amino acid is called N-terminal, and while the one at the right end is the last amino acid is called as a C-terminal amino acid. So in this picture you can see, so this is one amino acid, two amino acid, 3, 4, and the bond between this is your peptide bond 1, this is peptide bond 2, this is 3, and so on. So they will form a linear structure, the straight structures in this way they can show, and this one is your N terminal and this is your C terminal. So this is the first amino acid, and here you can have the last amino acid. So this is a linear structure of the protein known as the primary structure. Secondary structure. Higher levels of the protein structures are the result of the weak hydrogen bonds between the side groups of amino acid. Formation of hydrogen bonds depends upon the nature of amino acid in the polypeptide chains. So types of secondary amino acid. The first is alpha helix type. So as you can see, so here you can have the peptide bond, but along with that we can have the hydrogen bonds, and this shows the helical structure, means it is they are in the helical form. So by making the helical, they are being attached or they are being bonded with each other with the hydrogen bonding. So in this case, the polypeptide chain is coiled like a spring. Hydrogen bonds are formed between NH and CO of adjacent coil. They will form this is one coil, this is another, and the hydrogen bond is formed between the adjacent coils. The example of the secondary protein is keratin, which is present in our nails, in our hair, that is present in our body too. So that is the keratin, which is alpha helix type. Second is plated sheet type. In this case, the weak hydrogen bonds are formed between two polypeptide chains. So here you can have two polypeptide chains. This is one polypeptide, this is second, and in between they are being attached by the hydrogen bonds. That is, one is of NH group, that is amino, another is of the carboxyl group. And such type of proteins that is called as a fibroin found in the silk. So the weak hydrogen bonds are formed between two polypeptide chains, which together make the pleated sheet-like structure. So that is, the hydrogen bonding occur between the amino of one chain and carboxyl of another chain. Next is your tertiary structure. When the polypeptide becomes further stabilized by folding and the coiling, by formation of ionic bonds or the hydrophobic bonds or the disulfide bridges, the protein is said to be the tertiary structure. The folding occurs in such a way that the non-polar side chain get concealed, means chup jana, while the polar side chains, they are exposed. The example is of the enzymes like amylase, pepsins, and many other enzymes. So here you can see that is the hydrogen bonds between them. This is your hydrophobic interaction. This is the van der Waals forces, which is a weak forces. That is the ionic bond, and this is your disulfide bond, and they are in a coiling form with the concealed of the non-polar and the exposed of the polar end. Next is your quaternary structure. When the protein has many subunits or number of polypeptide chains, each having a primary, secondary, tertiary structure of its own, the protein is said to be the quaternary structure. The example is hemoglobin and insulin. So here you can see the four subunits are being attached and they form the complex protein structure, that is your quaternary structure of hemoglobin. So this is a quick recap of all the structures. That is primary, which is in the linear form attached by the peptide bond. This is your alpha helix structure like the springs with the hydrogen bonds in them. This is your beta pleated structure, beta sheet structure. Again, the weak hydrogen bonds are there. Tertiary, they are coiling and the folding with the polar and the non-polar ends. And the quaternary, they have the various subunits attached with the center, that is your hemoglobin. Next comes the classification of proteins. So based upon the physical nature, the proteins are of two types. Fibrous, in which the polypeptides are arranged in the polar bundles. Example is keratin, collagen, silk fibers are there. So they are present in the fiber form in the linear forms. Globular protein, in which the polypeptide becomes coiled and the folded, that is albumin, globulin and hemoglobin. Another classification is based upon the structure. The proteins are of two types. That is, simple proteins are those which are composed of only amino acid, that is histones and albumins, and conjugated or the complex proteins are those which have peptide chains and non-protein moiety called as cofactor. So in which the two particular chemical substances are present and if only the amino acid, that is your simple proteins. So here you can have a various names of the conjugated proteins, that is phospho in which the phosphate group is attached, glycoprotein in which the carbohydrates are there, nucleoprotein in which some nucleic acids are there, chromoprotein, lipoprotein, flavo and metalloproteins in which along with the amino acid groups we are having the non-protein moieties, that is called as the cofactors. So the next we have the very important topic, that is the functions of the proteins. So the first function of protein is enzyme. So it is very well known that all the enzymes are protein in nature. So what are enzymes? They catalyze the specific chemical reaction. Number 2 is the structural proteins, and the example is collagen. They strengthen and protect the cells and the tissue. Storage proteins, that is store the nutrient particularly abundant in the eggs, that is called as the over-albumin in the egg white. So egg white is having the albumin, which is a very good source of the protein, and the seeds are there in the corns. Transport protein, as we have already discussed, the hemoglobin is of a quaternary structure and the main function of the hemoglobin is to transport the oxygen in the red blood cells and move the specific substances, that is glucose, amino acid, across the membrane. Next is regulatory proteins. So some proteins are hormones, for example the insulin, that is also the example of quaternary protein. They control the expression of the specific genes, means they controls the various, regulating the various actions in our body like controlling the blood sugar level. Motile proteins participate in the cellular movements, for example actin and myosins. They are also called as the contractile proteins, which helps in the contraction and the relaxation of the muscles. Protective proteins, which protect our body or increase the immunity of our body, that is called as an antibodies, which defend against the foreign invaders. So we are having the 7 types of proteins which plays a very important role. So all enzymes are protein, but all proteins are not enzymes because they are playing the many other roles, that is enzyme, structural protein, for example collagen, storage protein, that is your egg white, transport protein, that is your hemoglobin, regulatory protein, that is your insulin, or sometimes you can find only the hormone is being written there. Motile or contractile protein, that is your actin and myosin, and that is your protective protein, that is known as your antibodies. And they helps to play a very important role in our immune system. This is all about our proteins, and our next lesson will be on the another macromolecule, that is a nucleic acid, that is your DNA and RNA. Till then, stay home, stay happy. Thank you. Have a nice day.